relax, come back at Okay, hello, welcome to the 18th ZOOMinar in the series on proteinopathies. Since this is the last ZOOMinar for 2020, I take this opportunity to officially thank the panel, Joan, Magda, and Bikash, for all the help. Without them, it's impossible to have been this successful. And all the great speakers, of course, they're extremely important. They give great seminars. And of course, the brilliant participants. Please continue to participate and with the same enthusiasm and energy level. Today, we have an exceptional researcher, speaker, Dr. Vladimir Uversky from the University of South Florida. Dr. Uversky obtained his PhD in biophysics from the Moscow Institute of Physics and Technology in 1991, and also obtained DSc, Doctor of Science in Biophysics from the Institute of Experimental and Theoretical Physics at the Russian Academy of Sciences in 1998. Then he moved to US, University of California, Santa Cruz, and later on joined Indiana University, Purdue University, Indianapolis as a research professor. Since 2010, he has been faculty in the Department of Molecular Medicine at the University of South Florida, where he works on various aspects of protein intrinsic disorder phenomenon and on the analysis of protein folding and misfolding. So here are some accomplishments Dr. Uversky has, which is really amazing and believable accomplishment. Dr. Uversky has co-authored over 950 scientific publications. This is until 3 days ago, maybe 50 more coming up soon, maybe. And since 2014, he is one of the highly cited researchers in the Clarivate list. He has collaborated with more than 12,500 colleagues from more than 2,750 research institutions in 89 countries or territories. He is an editor of a number of journals and he edited several books and book series on protein structure, function, folding, and misfolding. So with that, it's my pleasure to welcome Dr. Uversky. Dr. Uversky, the floor is yours.
Okay, thank you very much for very kind introduction. I'll try to start my presentation right now. And hope you can see my slides. Can you?
Yes, we can see it.
Okay, and I need to change pointer. Okay, so I will be talking about intrinsically disordered proteins, and I will talk about diseases. So before moving anywhere further, and so because of, can you see that? So my internet connection is unstable. I'll try to not to be disconnected. I will start with some acknowledgements. Rams mentioned that I have quite a few papers published, but it's not me. It's because I do have quite a few colleagues. And so early on, I decided that I want to have a kind of Facebook of my collaborators, and I started collecting faces of people whom I'm collaborating with. And it worked okay for some time. As you can see, I have quite a few faces here. I definitely don't have time to explain how they ended up in this Facebook. All of them did something. We worked together. We probably have, not probably, very highly, with very high probability, we have joint publications. And it's almost approaching the end of my Facebook. Probably this is last slide. Yes. And everything worked okay. So before 2016, and in 2016, I published a paper, which collected, as you can see here, 2,427 courses. So definitely, I lost hope to collect faces. And even if I kept this hope for some time, so last year I published another paper with 1,500 courses. Now definitely I cannot have. I don't have any possibility to collect pictures of all of these colleagues. So as Rams mentioned, I collaborated with almost 13,000 colleagues from quite a few research organizations, probably in almost all countries which have scientific research. So this is my interactome. So, and so definitely stuff which I will be presenting today, so will not be possible without collaboration and contribution from all of these colleagues. So I'm definitely very, very thankful to all of them. So now, two very important messages. If you do like what I will be talking about today, so keep in mind that it's not me, it's all of these people. So if you don't like, so don't blame them. So it's not them, it's me. I was not able to deliver message in the right way. Okay, so what I will be talking about today is shown here. So it's an outline of my talk. I will introduce intrinsic disorder very briefly. I will show you how intrinsic disorder can be found. We'll discuss a very important concept, protein structure function continuum, and we'll show you how disordered proteins are related to neurodegeneration and how this structure function continuum is converted into involvement of all of these diseases, all of these proteins in multiple diseases. Introduction of intrinsic disorder. If you open any textbook and start to read about proteins, what you will see there, you will see this classical structure function paradigm according to which amino acid sequence is able to fold into specific structure, and this specific structure will be responsible for protein function. So we do have very important information flow, so starting from sequence ending with function. So and so this is one key idea which was proposed by Emil Fischer in 1893, and it was elaborated in order to introduce some mobility and flexibility in the form of induced fit. So, but altogether, all of the stuff is based on assumptions that so we do have a unique structure which is responsible for unique function. So this is an extremely interesting and important idea, and I'm considering it as a big bang of protein science. So because as soon as this idea was proposed in 1893 by Emil Fischer, so a lot of people start to work on proteins and they start to elaborate all of these branches of protein science which are shown here. As a matter of fact, so by the end of last century, vast majority of our knowledge about what protein is and what protein can do was derived based on this structure function paradigm. However, so many proteins were found not to follow these major rules, and so they were taken as an exception. So quite a few proteins were shown to be not having specific structure, but still possessing very important functions. So these proteins were observed starting from as early as 1946. So, but people mostly consider them as an exception from a general rule of structure-function relation, and so they were more or less ignored. At the end of last century, so a few groups, including mine, so came up with this interesting idea that so these non-foldable proteins are also known as intrinsically disordered proteins and also known by about 50 different other names. So they're not exceptions, and they could be abundantly present in nature. So, and as a result, now we do have this interesting representation of potential fate of a polypeptide chain inside of cell. After biosynthesis, a protein can fold. And so this is structure function pathway. And so people were analyzing this stuff for more than 100 years. So some proteins can misfold. And so we have misfolding pathway. And so this ZOOMinar series is talking about this misfolding correlation. Curiously, misfolding and aggregation were known for a very long time, but they were mostly ignored by researchers because everybody was trying to work with structure. And misfolding and aggregation wasn't giving you structure, wasn't giving you a possibility to analyze structure-function correlation. And about 25, maybe 30 years ago, a correlation was found in between misfolding, aggregation, and human diseases, and situation changed because now, so we can talk about molecular mechanisms of diseases. So in addition to this folding and misfolding pathway, we do have another very important cellular pathway, protein non-folding. And here, protein don't have structure. They don't fold to different degree, and I'll talk about this stuff in a second. So, so they don't have rigid structure at the end. So, but they are biologically active. So what is also important, so these pathways, they are not dead ends. So because, so sure, here you want to ask something? Go ahead. No?
Vladimir, we'll take the questions at the end.
At the end, okay, that's fine, absolutely, it's okay. So we do have this, so none of these pathways will be dead end. So because folded protein can misfold, and we know about the stuff very well. Some misfolded protein can be brought back to folded state, so using chaperone and other machinery in the cell. And folded protein can undergo partial unfolding or complete unfolding in order to become functional. And there is a very interesting phenomenon related to this transition from structure and lack of function to disorder and presence of function in a given protein. And also disordered protein can misfold and go back. So all of this stuff goes to circles. So, so this also will give us a very interesting perspective on what this disordered protein look like. So they are highly heterogeneous from structural viewpoint. So therefore, I'm discussing them from viewpoint of structural spectroscopy. So what do I mean by that is shown in this slide. So when we just start talking about intrinsic disorder, we considered just two categories of proteins: structured proteins, ordered proteins, rigid like rocks, and this is actually major subject of protein science for a long time. And so completely disordered proteins is noodles, so which also have biological functions, but which don't have any structure. So at the beginning, it was very necessary to find such kind of proteins, which looks like noodles, which don't have any structure, but still have very important biological functions, because it was necessary to oppose this rigid view of protein functionality. Almost immediately start to be clear that so this red, blue, or black and white presentation of protein structure and disorder is a misconception, because what we have in reality, we don't have this just two colors. So we have this continuous spectrum, in which we have highly structured protein on one side of spectrum and highly disordered protein on another side of spectrum. And we do have this broad set of differently disordered proteins. So protein can be disordered to different degree as a whole, as entire protein, or any protein part can be disordered to different degree. And as a result, we do have this continuous spectrum of different conformational states and different structures of proteins. In reality, not only IDP, but any protein can be described as a combination of differently folded regions. Some regions will be called fold-ons. So these are independent foldable units, which are not domains, which are smaller than domains. So because a single domain protein might have quite a few fold-ons, at least more than one. In addition to fold-ons, we have inducible fold-ons. So this will be disordered regions, which are disordered by themselves alone, but when they're interacting with specific partners, or if they're located in a specific environment, so they will be able to fold. This will be inducible fold-ons. We'll have also inducible morphine fold-ons. Sorry, I didn't list it there. So morphine fold-ons will be foldable regions which can fold differently upon interaction with specific partners, and I will mention this stuff once again so later during my talk. In addition, we also have non-fold-ons. So this will be non-foldable proteins or non-foldable regions, which never will get any structure and which will stay disordered all the time. And there is a pretty large set of functions, so which will be associated with this lack of structure. We might have semi-fold-ons, which are almost in semi-folded state. And also we can have unfold-ons. This is what I already mentioned. So these are regions of protein, so which are structured but not functional, and which need to undergo this ordered disorder transition in order to become functional. So it's next question will be how common these proteins are. So when I just started to work in this field, I started with just one protein, and so I convinced myself that so we do have new phenomenon. So when I experienced protein number 3, and if somebody would ask me a question at that time when I was just starting this work in this field, How common these proteins are, I would say, so they are more common than just exceptions. But they're not very common because we did not find too much of information about these proteins in literature. And I would be completely wrong, because when we elaborated computational tools for rather accurate prediction of intrinsic disorder, and when we applied these tools to different proteomes, we got these amusing results. What I'm showing here, so this is organisms which we analyzed, archaeobacteria, single-cellular and multicellular eukaryotes. This is number of proteins in different organisms, different number of organisms in this in any given category. So this is a range of their proteomes. This is an average proteome size. And as you can see, so we do have this very interesting correlation between increase in protein organism complexity and a number of proteins. More complex organisms will have more proteins. And also we did this three very simple type of analysis. So for finding intrinsic disorder, analysis number one is really very, very simple. We applied computational tools to see how many residues altogether in any given proteome are predicted to be disordered. And so we got these values from about 10% to about 45, 47%. And then we were told that having these disordered residues which are spread through entire sequence, maybe it's not very biologically relevant. So what we need to look for disordered regions. And we defined disordered region as region which has more than 30 predicted disordered residues in a row. And when we redid this analysis, we found that from 10 to about 70% of proteins in any given proteome, so we'll have these long disordered regions. So we also have computational tools which can, it's kind of binary predictor, yes, no, so which can tell you if your entire protein is expected to be structured or disordered. And if you apply these tools, we can find that from 3 to about 40%, in any given proteome could be completely disordered. So it's a lot of disorder for sure, and it definitely shouldn't be ignored. It should be taken very seriously into account. So this is same data, but shown not in the table, but in the plot. So what I'm showing here is an average fraction of disordered residues, dependence on proteome size. So we do have simple organisms shown here. So it will be bacteria green and archaea blue circles. And I have more complex organisms, which will be single-cellular and multicellular eukaryotes shown by brown squares and pink triangles. And also I have viruses. Viruses will be very different story. I don't have time to talk about this stuff, but I will just mention very interesting observation here. As you can see, in order to go from simple cell to more complex cell, what nature is doing, it's using increasing proteome size. But not only increase in proteome size, but also pretty dramatic increase in amount of intrinsic disorder. More complex organisms will have more disorder, and increased disorder content is really very crucial. So for increasing complexity of organisms, which means that intrinsic disorder has some functions which are really very crucial for more complex organisms. Yeah, definitely. Disordered proteins are very common, and they need to be present because they're functional. This brings us into this very interesting. If you will exclude intrinsic disorder from consideration, if you will consider just structure, so we will have our classical structure function pathway amino acid sequence. We'll solve following problem, we'll get specific structure, and we'll get specific function, and this function will be described in terms of lock and key or induced fit. So now, if you will add intrinsic disorder at any given moment of protein life, what protein can do now is dramatically increased. So a functional repertoire will be very, very extended. In some cases, structured protein can be partially disordered part-time, and we'll get into new specific structure. We have conformational speech. many structured proteins need to become disordered, and it will be very crucial for a set of functions which are listed here. It's not a complete set of functions, it's just illustrative examples. And this blue pathway will be starting from structure. In addition to appearance of disorder on some moments of protein life, so from structure. So we also have this another pathway in which amino acid sequence is not worrying about folding, and it doesn't exist as structural, as unique structure. It exists as a very flexible conformational ensemble. So it will be quite a few functions which will be associated with this entropic activities, very crucial for such kind of disordered proteins. But some of them will be able to fold upon interaction with specific partners, and we do have another set of functions associated with this disorder to order transition. So once again, what is important to take out from this picture here? So when we are adding intrinsic disorder, when we are taking intrinsic disorder into consideration, so we are dramatically increasing of what protein can do. So it's quite clear that disordered proteins are almost incompatible with catalysis. And in reality, I do know probably 5, maybe 6 enzymes, so which are molten globule-like conformation, but which are catalytically active. So and it's more exceptions than rule. But disordered proteins are very common, not in catalysis, but in some other activities which are very crucial for proteins, and this will be regulation, recognition, signaling, and ability to interact with multiple partners. So all of this stuff will be very crucial from your point of the intrinsic disorder. So, and question is why? So why disordered proteins will be very good in signaling? So, and I will give you a very brief description of a few observations of how it works and why it works. So first of all, so signaling interaction will be characterized by 2 very kind of opposing criteria. So signaling interaction should be specific. So because you need to transmit your signal into right target. So, but also this interaction should be weak. So because signal should go in and on and off all the time. So how you can handle this stuff? So you need to have highly specific interaction combined with low affinity. So it's impossible from viewpoint of structured proteins for which affinity and specificity are synonymous. Each time when I have a highly specific interaction, so by default I have very high affinity, or vice versa, if I have high affinity of interaction, so interaction will be highly specific. However, if we do have this situation shown on this slide here, so when we have disordered protein, which is able to interact with structured protein, and will be able to fold into specific structure, so in specific binding group of binding partner. We do have specific complex formed at the end. So because the protein was disordered to start with, part of energy will be spent for folding, and as a result, this complex will be weaker in comparison with structure-based complex. Very interesting note here. Weakest interactions in protein world will be in between disordered protein and ordered protein, or in between 2 disordered proteins. But strongest interaction in protein world also will be in between disordered proteins. So because you have possibility to have conjoint folding and binding, and you can create very sophisticated, very intertwined structures, so it will be extremely stable. So this is why disorder is crucial from viewpoint of signaling and complex formation. IDPs can form this polyelectrostatic type models, and I'll use my fingers here. So this will be such kind of interaction. So you don't have any specific contact formed. So what you can have, you have this protein plane staccato model. So in which multiple binding sites on disordered protein will be able to interact with one binding site and structured protein. And so each of these binding sites has equal probability to interact. And as a result, at any given moment, for in ensemble of structures which we have in solution, so we'll have all possible combinations, and as a result, we cannot see any specific interaction, structural changes associated with specific interaction. But what can we see instead? We can see pretty stable binding. And such kind of interaction is possible, but also such kind of interaction is possible when both proteins are disordered. They also can be engaged in such kind of touching interaction. As a result, so we do have induced folding, we have proteins playing staccato, and we have Mr. Burns with his excellent statement here. So all of these interactions will be based on disorder. I forgot to mention here, so this is really very interesting model here. We have structured protein, so interacting with disordered partner. Disordered partner has multiple phosphorylation sites. Each of phosphorylation site has equal probability to be bound to partner. And so strength of binding is directly proportional to degree of phosphorylation. If you are starting from 9 phosphorylated serines and threonines as its normal state of binding partner, you have strongest interaction. And if you take out one by one phosphorylation sites, so from 9 to 5, so you have proportional decrease in affinity. And if your binding partner has less than 5 interactions, it's a shutdown mechanism, so there is no interaction anymore. So you do have possibility to have rheostat-like interaction, which is highly adjustable by phosphorylation, so because of intrinsic disorder. Now a few words about application of intrinsic disorder for alternative splicing. I don't have too much time to go through this stuff in much detail, therefore I will be very brief. What is alternative splicing? Everybody knows. We do have eukaryotes, we do have introns and exons, and maturation will lead to removal of non-coding regions, and we do have our mature mRNA, so which will be canonical isoform, which will be coding into a specific polypeptide chain, which is able to fold into specific structure. This will be normal splicing. Alternative splicing means that, so in addition to non-coding regions, some coding regions can be taken out. As a result, we have an alternatively spliced mRNA, which will produce an alternative polypeptide chain with a known fate of folding. So if we are considering just structured proteins, for which it's quite known that almost any piece of sequence is crucial for folding, because we do have this folding code in which amino acid sequence knows how to fold into structure. If you take part of this code out, remaining part will not be able to fold. But somehow alternative splicing is not killing organisms. Definitely what it will be if, this foldable protein will be losing a very important part from its globule, it will not fold. It will be misfolded, it start to aggregate, you have this conformational disaster. So somehow you have a very interesting contradiction here. Most probable output of alternative splicing from viewpoint of proteins will be losing structure, getting some problems with folding and conformational catastrophe. But nature is using alternative splicing very, very intensively. 75% of proteins in human, we'll have alternatively spliced isoforms. If you'll consider only proteins which have exon intron this mosaic structure, 95% of these proteins will have possibility to have alternative splicing. So how come do we have this combination? And answer is very simple. Stuff which is alternatively spliced in mRNA, will encode mostly for intrinsic disorder. We are taking any piece of no structure, remaining part of no structure, we'll be able to survive. So we still have about 20% of proteins for which alternative splicing will happen inside of structured regions. And what we can have in this 20% of proteins, it will be surface loops, independently folded domains, or 2 to 3 residues from protein globe, from protein core. This will be present here. And in all other cases will be associated with intrinsic disorder. And but it also has a very important role from viewpoint of looking on functionality. We do have, for example, as it's shown here by breast cancer protein. It has a structured N-terminal domain, a structured C-terminal domain. It has very long disordered region. It can interact with multiple partners. It has quite a few alternatively spliced isoforms. And so shaded areas will show you how alternative splicing will affect dramatically what this protein can do. So, and major conclusion from this picture here will be, so combination of alternative splicing with intrinsic disorder is crucial from a viewpoint of increasing functionality of protein. So we are not killing structure because we don't have structure, but we are able to extend functionality of protein to a very large degree. So now I need to accelerate because time is short. Disorder is crucial for post-translational modifications. We have about 520 kinases, so which are able to phosphorylate almost each and any protein in our organism, and many proteins will be able to phosphorylate in many different places. So we have lesser number of phosphatases, and all phosphorylated proteins can be dephosphorylated by kinases. So which means that for these proteins, we don't have, we cannot apply in an easy way lock and key concept because we do have too many keys for one lock. And I'll probably skip this stuff. We'll go directly here. So for phosphorylation, acetylation, ubiquitination, and many different other types of post-translational modifications which are based on catalytic activity of specific enzymes do these modifications. Intrinsic disorder is crucial because sites which are modified need to be accessible, need to be highly flexible, need to be able to adjust to active site of enzyme, and so all of the sites will be located inside of disordered regions. Same problem is related to protein-protein interaction networks. So this is one of example of protein-protein interaction networks. So we have quite a few proteins which will be engaged in multiple interactions. So it will look like these flowers. These are so-called hub proteins, and hubness is not compatible with lock and key. And intrinsic disorder is crucial for protein-protein interactions because many hubs will be completely disordered. If hub is not disordered, it will be interacting with disordered binding partners, similar to what is going on with kinases and partners of kinases. So as a result, disorder will be crucial on one way or another, but still will be present. We don't have rigid hubs interacting with many rigid partners. So this is another very interesting idea related to what disordered protein can do. This one-to-many binding scenario, and P53 is a very nice example of this scenario. Once again, I don't have time to go through this stuff in much detail. So only one idea here. So in PDB, you can find more than 30 different structures of P53. So none of the structure will be full-length protein, so it will be pieces of P53 co-crystallized with binding partners, as it's shown here, with exception for DNA binding domain. So so you can see that these differently colored pieces, so we'll be interacting with cyan clouds, which will be binding partners, and most important part probably will be here. It's CTMNO part of P53. So which is able to fold into alpha helix, into beta strand, or into irregular structure of different configuration. So it's exactly the same sequence. So depending on environment, it falls into different structures, and it's able to control and regulate at least 4 different pathways. In reality, much more than 4 different pathways. So intrinsic disorder is really very crucial from this viewpoint. And it brings us into this very important protein structure function continuum idea, in which protein which has differently folded regions, fold-ons, unfold-ons, and everything else which is shown on this slide. And each of these differently folded regions is able to do very specific functions. And which means that so for any given protein, we can have quite a few different functions because of its disorder to different degree. And as a result, we don't have just structure function, so we do have structure function continuum. And I will skip and I will go over there. So once again, instead of one gene, one polypeptide chain, unique structure and function. So what we have a gene, multiple polypeptide chains produced by alternative splicing or post-translational modifications or by some other stuff, and intrinsic disorder. Instead of unique structure, we have conformational ensembles, and we do have quite a few different functions associated with one particular protein. So, and I don't have time to talk about it. So So it gives out this firework representation of protein functionality. From one gene, we do have quite a few different functions, which are shown here. So associated with alternative splicing, mutations, possible modifications, or combinations of all of these factors. So, it actually brings us into another important point about how intrinsic disorder is controlled inside of cell. And this is really a very important point because we need to know what is going on there. So, so disordered proteins are controlled differently in comparison with structured protein at any given moment of their life, from transcription to translation to degradation. So they are controlled in different ways. So you do have special machinery which handles structured proteins and disordered proteins in different way. And so this machinery helps disordered protein to stay in good shape. So however, if something is not working right, so we do have this D-square problem, which is disorder in disorders or intrinsic disorder in human disease. If you have problems with regulation of IDPs, so like problems, mutations, chromosomal translocation, alternative splicing, and problems with expression or non-genetic factors, aberrant degradation or wrong process modification or defective trafficking. So all of the stuff can cause proteins to misbehave and be engaged in some pathological processes. Chromosomal translocation is bringing two genes together and removing central pattern instead of two different proteins having these hybrid proteins, so which are very common in cancer. Alternative splicing instead of normal structure, so you can have some wrong structures, and you have this conformational disaster which I discussed, talking about alternative splicing. So it's also very common in different diseases. Aberrant phosphorylation modification are directly related to neurodegeneration. So you have tau protein and problems of hyperphosphorylated tau protein. Wrong degradation, it will be amyloid beta in Alzheimer's disease. So now I will talk about so how you can go from multifunctionality into multi pathology of disordered proteins, and I will use Parkinson's disease and alpha-synuclein as an example. Parkinson's disease, you know that it's second most common neurodegenerative age-related disorder. It's an environmental disease which is associated with exposure to pesticides and metals. It's associated with loss of neurons in substantia nigra, and it's associated with accumulation of Lewy bodies and Lewy neurites. So you know this stuff, I will not talk about it. I'll just show a couple of slides and let's Lewy bodies, Lewy neurites. So what is important, so major player or one of major players in Parkinson's disease is alpha-synuclein. And in 97 it was shown there are a few mutations in alpha-synuclein which are related to familiar forms of Parkinson's disease. And it was a beginning of analysis of correlation between this protein and Parkinson's disease. But what happened, alpha-synuclein is disordered. It can form amyloid fibrils depending on in which place of brain, so this aggregated material will be accumulated. So we do have different form of accumulation and different forms of neurodegeneration. So this will be probably most important slide related to different pathologies. These are different synucleopathies. In 97, it was just Parkinson's disease. Now, I think it's not complete table. I think it's probably half of table which need to be shown here. So this will be different diseases, so which are associated with misbehavior of alpha-synuclein. So why it's so? Alpha-synuclein is disordered protein. It has multiple isoforms. It has, as shown here, it has multiple possibilities for be modified. It interacts with a very large number of different partners. It's disordered by prediction. It's disordered by computation. It's a very important half protein interacting probably with more than 400 proteins. So it's a protein chameleon, which is able to fold into different structures depending on the environment, more or less similar to what I showed to P53, but now we are talking about whole length protein here. and it's still considered as protein with unknown function. So which is actually amusing to me. So we do have about 20,000 publications related to alpha-synuclein, and vast majority of these publications will start in introduction stating, so this is an important, maybe intrinsic disorder will be mentioned, protein with unknown function. So, and then it will be discussion of how this protein is related to some functionality or some other functionality and some pathology. So why protein with unknown function? So this is a misconception coming from this overall idea that protein need to have specific structure in order to be functional, and that one protein need to have one function. In reality, so alpha-synuclein represent a very nice example when you have one protein with multiple functions. It's not one function, it's not unknown function, it's multiple different functions. So, and so this is an octopus view of alpha-synuclein functionality and pathology. So when it's disordered, it can do a lot. So by itself, when it's aggregating, it can do more. So because now instead of one octopus, you have many, and each of them can do something. And so definitely you need to apply a proteoform idea here, and I forgot to mention, I'm sorry. So proteoform, it will be all different protein forms which are produced from one gene. And these different proteoforms will be produced by alternative splicing, post-translational modification, or will be present because of intrinsic disorder. If you will apply them to alpha-synuclein, you will have possibility to see malfunctionality, multifunctionality, multipathogenicity, so multiple synucleopathies. And so it will be also connections to multiple different other diseases. So, and polymorphisms of aggregated forms, presence of strains and related to transmission, and a lot of other stuff will be easily explained if you will take intrinsic disorder into account. So, and a few last slides will be to show that alpha-synuclein is not an exception. So we have this triad, synuclein, amyloid beta, and tau. So they're related to neurodegeneration. Some of them will be found in specific diseases. So, but a combination of them can be found in joint diseases, as it's shown in this slide. So I gave you pretty brief description of alpha-synuclein, and tau. It's same story. It's completely disordered, and amyloid beta is also completely disordered, and their connection and involvement in different diseases. So we'll be coming back to their lack of structure and ability to be differently structured in different environments, and because of this disorder, being able to engage in multiple different functions. So so this really will be a couple of my last slides. So now we have intrinsically disordered proteins, so which represent a new concept in protein science. Now we are able to explain a lot of stuff which wasn't explainable, so from viewpoint of structure function correlation, because so we do have a lot of protein activities which are not based on structure, which are based on lack of structure, and this stuff need to be taken into account very seriously. So, and my concluding remarks are very simple. So we do have quite a few disordered proteins in different proteomes. So they are very common in signaling. So intrinsic disorder is crucial for protein structure function continuum. So these proteins are tightly controlled in the norm. If they're not controlled, we have quite a few different diseases. And because of they're multifunctional, they also could be, each of them can be related to multiple diseases. So because of lack of structure. Thank you for your attention.
All right.
And I'm ready for questions.
Thank you very much, Vladio. Let's see if there are questions. Please raise your hand or post your question in the Q&A folder. It was a great talk, Vladio. Thank you very much.
No questions. Everything was clear. Well,
No,
two explanations. Yeah. So either it's completely boring or everything is clear.
Okay, Magda, go ahead.
My question is, how do you I was trying to envision how do you discriminate from the different foldons, unfoldons in your predictions? Because you talk that some regions are like foldons, some of they don't fold, some of them partially fold. I was kind of trying to figure out how you're doing it. It's just assembly of the sequence base or like multiple other factors.
Conclusion about conclusion about different types of foldability. It's more or less analysis of data. So in prediction, you can easily see what you can see disorder. So because we have computational tools which are showing proteins which are disordered, you can easily see structure. So, but also there are some regions. So let's go back to P53. It will be a good example, actually. Yeah. Almost there. Yeah. This one. This is a typical prediction of intrinsic disorder. So for a protein, and so you do have a region which is predicted to be disordered, but it also has a spike of structure. What it tells you that you have a region which potentially can fold. So, but it will not be folded because it's located inside of disorder. So it doesn't have enough energy to be folded by itself. So, but it can fold if appropriate partner will be found. And as you can see, so these pieces will be co-crystallized with binding partners. So same is here for C-terminal part. So by looking on this disorder profile, I can say, okay, so this will be structured domain, so this will be disordered domain. So it will have probably fold-ons and something else. This will be disordered region, so which has some inducible fold-ons. So this will be able to fold upon interaction. So as far as unfold-ons, I'm not sure that so we do have any computational tools for predicting it. So unfortunately, we don't have too many examples so far for developing tools. Did I answer your question?
Yeah, that's perfect. That's a really nice way to look at it, because usually when we look at the predictions, oh, this is not folding, and we just move on. But it's really a nice nice thing to keep in that. My other question, which I'm thinking, you were talking quite a bit about post-translational modifications and like how they fine-tune the ability of the proteins, like to fold, unfold, and stuff like that. Do you have a database in which you just do the same, or you maybe edit to do the same predictions, but in accounting, let's say, if the user wants all like phosphorylations or acetylation and other stuff like that. Have you thought about something like that?
We do have, it's actually a database. So it's G2P2. So this database will give you a very nice picture. So if protein is there, so it was designed quite a few years ago, and originally it included all proteins from all annotated proteomes. So, but annotated proteomes are increasing in size. Even protein in human will be kind of more and more annotated. It doesn't cover everything. So, but if you're lucky, what you will have in this D2P2, you will have a profile which will show you distribution of intrinsic disorder. It will show you presence of conserved functional domains. I will show you presence of these molecular recognition features. Will be these pieces, this stuff which are able to fold upon interaction. And it also will give you position of multiple phosphorylation or acetylation or different type of post-translational modification sites. And it's an interactive picture. You can click on all images which are present there, and it will give you some information about the stuff. So probably this will be the closest stuff to what you want to look at.
Yeah, thank you. Because many of all this relationship between the disorder and disease, as you alluded, it's really tied up to the, like in many occasions, to the post-translational modifications, and that's really helpful.
Okay, thank you, Magda.
We have people on
Vijay,
the line.
Another possibility. So yeah,
Go
Magda,
ahead.
one more comment. So easiest way will be to contact me.
No, go ahead. Go ahead. You
Okay,
continue.
so let's go for other questions.
Yeah. No, you
No,
finish
what
the
I
comment.
said that so easiest way in this case will be to comment. Yeah, I'm done. Yeah.
Okay, so Vijay, go ahead with your question.
Okay, can you hear me?
Yep.
Hi, Vlad.
I can hear you. I cannot see you, but I cannot
Oh,
see you. I can hear
oh,
you.
looks like
Yeah,
my video
hello.
is also.
Oh, I can see you now. Excellent. Oh, nice
Okay,
to see you.
great. How are you?
I'm fine. How are you?
Good, good. Well, I just have wanted to ask you about your take on some evolutionary aspects of this disorder, right? I mean, if you think about and from an evolutionary basis, I would imagine that the disordered proteins would be likely to have present before the folded part came in. But if you look at this, is your spread of the presence of disordered proteins in the higher order species, it seems to be more, which is sort of counterintuitive to this theory. What is your take on it?
It's not. It's not. So because we are considering here two very different time frames. So you're absolutely right. Early on in prebiotic or early biotic time, so definitely first proteins were disordered. So there is no way for nature to create structure. So definitely, and I can go into a pretty lengthy discussion of what kind of evidence do I have for it. So it's not direct, so it's kind of imaginary, but still from our viewpoint, it's pretty solid. So you're absolutely right. Early on, so we had a lot of disorder. What it will be first function for such kind of proteins. So depending on if you do believe in RNA world or you don't believe, if you do believe in this case, an explanation is very simple. So first function of protein will be RNA chaperone. So they will be wrapping around ribozymes, will be helping ribozyme to be stable and do whatever is necessary. So then, so why we got proteins? So because we have 20 residues. And we do have very high level of physical chemical diversity. So we are able to create very sophisticated active sites. So now we are able to catalyze. Yes, we are able to, we can create some catalysts. Evolutionary, very big advantage of cell over another cell will be from viewpoint of independence. If I'm very selfish, if I'm able to produce everything for myself inside of myself, I will be having more chance to survive. So which means that so first move into inside of evolution so was related to pushing into structure. So we moved from complete disorder early on into very large amount of structure in simple organisms. So and then we had a transition from simple organisms into more complex organisms. Now we have different organelles. We have multiple tissue. We have multiple cells talking to each other. Now we have different levels. We have re-engagement of intrinsic disorder. It's rediscovery. So we started from disorder, ended up with structure, and moved into more disorder. So with evolution. Make sense?
That is the same logic applies to this low complexity domains as well, right? So
yeah, of course, absolutely. Yeah, but low complexity it will be a reflection of intrinsic disorder anyway. Yeah, absolutely.
Thank you.
Sorry, Danilo Miladi, go ahead with your question.
Hello.
Danilo? Yeah, go ahead.
Can you hear me?
I can hear you, and I can see you also. Yeah.
Okay. So first of all, nice talk. Congrats. Very interesting lecture.
Thank you.
I see that there are two key parameters in this story. The first one is the percentage of ordered structure in a protein, and the second issue is the mechanism of protein folding. I'll try to provide some examples. First, the most simple one is the all or none, classical all or none mechanism of protein folding. Or it is possible to have a definite number of intermediate states along the folding pathway. Or even more complicated, we have a very large number of intermediate states that could be virtually infinite number of intermediate states. And this is the normally named the Downing folding mechanism. So which is almost unexpectedly neglected. But my question is, in your opinion, what is the connection between these two key parameters that describe protein folding? Other structures and protein folding mechanism.
Okay. Okay. So for other proteins, so you can see multiple intermediate states kinetically. So not for all, but for many. And it could be quite different kinetically populated intermediate states. When you're starting from unfolded state and you're observing protein folding process. So for many globular proteins, you can change environment and you can stabilize different partially folded intermediates. And it could be quite a few of those also. Originally, people were debating just about molten globules, and pre-molten globule was introduced, and highly ordered molten globule, and probably much more in between. But so here, situation is different because it will be same sequence, but in different environments. So you're changing environment, you're populating different partially folded intermediate states. With disordered proteins, situation is quite different. So because what we have same environment, the physiological environment. So, but somehow some proteins will be folded to different degree. And this is a reflection of different sequences. So we do have some sequences which are not able to fold at all. I did not mention this stuff here, so but my journey into intrinsic disorder started with a protein which was brought to me in '93, long time ago. So guy came to my lab saying, I have a funny protein here, so you can boil it a week. So, but as soon as you cool it down, it will be completely active. So, and I cannot measure concentration of this protein. And I'm looking at him and saying, okay, so what are you talking about? Is it a protein from thermophile? And he's saying, no, it's human protein. And I'm telling him, no, it's impossible. You cannot have human protein which sustain a week of boiling, so without losing any activity. So, so he showed me a sequence. So the name of this protein is proteimazine alpha. It's 110 residues, and 70% is glutamic acid and aspartic acid. It doesn't have any aromatic residues. So therefore, you cannot measure concentration, yes? And so it will be highly unfolded because you have very strong charge repulsion. So this will be a very extreme case, probably one of most extreme cases for completely unfolded protein under physiological conditions. But now you can have different combinations of all of this stuff. So a very important point, which is ignored by many people, not only stuff which you mentioned, we might have multiple gazillions, unlimited number of different intermediate states. So, but another very important point, which is completely missed by many people, we don't have homogeneous structure. Even well-folded protein is highly heterogeneous. So you do have regions which are more folded, you have regions which are less folded, and this stuff. So I know why it's taken as highly homogeneous, because it's easier to model. In this case, we have structure, we can look at it. So, but reality is much more complex.
Thank you. Thank you very much.
Okay, Bina, go ahead.
Hi, can you hear me?
I can hear you, yes.
Yes. So my question is, is there any evidence or information that the intrinsically disordered region of a protein has either an active or a passive role in folding of the ordered part of the protein?
Yes. You're familiar with pre-pro proteins, yes? So you have
right.
precursors which will have some regions. And it was shown that so for many proteins, so these pre-pro regions, they're not structured. So, but they're really very crucial from viewpoint of folding. Another very important role of such kind of disordered tails, so will be keeping your protein soluble. It was a very interesting. Right. It was a very interesting study published in 2004. I don't remember the name of protein, but I do remember story behind this protein. So people tried to crystallize it, and they failed. And they did disorder analysis, and they found that so last 100 residues of protein are highly disordered, And they decided that this stuff which is preventing from crystallization, it took 100 residues off, and proteins start to be completely insoluble. It's fall out of solution.
Right.
So, and they were able to fix this problem by keeping some piece of this tail present. So which means that, so yeah, you do have, and you know, so to be completely honest, many chaperones will be highly disordered. And what you're asking about, so it's exactly the same stuff. Now we don't have external chaperone, we have internal chaperone. We have the division which is helping potentially foldable part to be folded, yeah.
Thank you.
Okay, so it's 11 o'clock. Let us formally close this session and we can continue the informal session. So let me take this opportunity to thank the speaker and all the participants throughout this ZOOMinar series. And on behalf of the panel, I would like to thank all of you and wish you happy holidays and a happy New Year as well. We'll see you in the New Year.
Well.
The informal discussion will continue. So Vladi, you're not leaving. We are going to continue to discuss.
I'm staying, yeah, sure.
So anybody who's wishing to join the panel, just raise your hand. I'll enable you to join the panel so we can have informal discussion. So Vijay is coming back. I'll just promote him to the panel. So Vlad, let's see here. There are more people wanting to come. Eliezer is coming. So what happens to the enzymatic degradation process that would how can a IDP stay as an IDP forever when there are enzymes around? So what kind of factors are competing other than phase aggregation, phase separation? Okay. Could you comment on that? Yeah.
So you gave a good answer, phase separation.
Yeah.
So you can imagine.
Other than that, I know phase separation is going to save them, but there are proteins which also don't phase separate out and remain as IDP. How do
phase separate? Yeah. So first of all, I don't believe that that all proteins are equally freely floating through entire cells, so they're compartmentalized. So one of possibilities will be, so compartmentalization works in this way that so disorder is not seen unwanted proteases. Number one. Number two, in vitro, all of these disordered proteins will be highly digestible. So it's not a question. And many proteins during recombinant production, if they're disordered, they will be highly digestible. So, but it's not happening all the time. So, and one of explanations, as I mentioned, so they're located in different places. Other explanations, they're engaged in interaction with partners and then somehow folded. One more explanation is phase separation, and even better explanations, they're located in places at which proteases doesn't exist. So you know, for example, if you take our eyes, yes, so half of proteins in our eyes are highly disordered. So what is going on inside of eyes? So we don't have any change of proteins. All proteins which we have inside of our eyes, so they're exactly the same as we got them so during embryo development. So which means that the half-life of proteins inside of eyes, it will be our lifetime or even longer. So if somebody, when I will die, somebody will take my eye and keep it. So proteins still will be disordered over there. So it's specific environment over there. Another very nice example will be elastin, highly disordered protein. So, but it forms bundles. So it starts to form very, very complicated structures. And when it's formed, so half-life of elastin is 70 years. So it's definitely depends on how the stuff is handled and where it's located.
Yeah, maybe elastin may be an exceptional outlier because it's more like polymer-like. It has local structure as well. So, okay, let's take the question from Eliezer and then we'll go to Vijay. Eliezer.
Yeah, hi. Hi, Volody. Good to see you.
Nice to see you.
I was a little late. Good to hear you. You're right. I was a little late for your talk because of time difference and keep forgetting it starts at 7 here. But you talked about, there was a question in the audience about regions outside the folding domains, how they help folding. Can you comment about how they help regulation? in addition
So,
to
but
folding.
it's same story. If you will assume that the vast majority or many post-translational modifications will be located in such kind of regions, now you have your regulation mechanism. So because of they're able to fold differently upon interaction with different partners, you have another regulation mechanism. So this stuff which I'm showing here, for example, on my slide now, so this C-terminal part of P53, so it's able to fold differently. So you do have probably unlimited possibilities how intrinsic disorder can be used for regulation. Alternative splicing included, post-translational modifications, and all of this.
And so how far should they be from the folding domain?
Or I think it doesn't matter. So, yeah, so what is good actually here, I will make this joke, but this is truth. I can answer any question, and I always will be right. So it's not because of I'm so smart. It's because of intrinsic disorder is so heterogeneous.
Yeah.
So
Okay.
you always will be able to find a case which will be supporting that I'm right.
Okay. Thanks.
Vijay, go ahead.
I'm sorry, I just can you hear me?
Yeah, yeah.
Okay. I can see you.
Yeah,
It's
that's
better.
good. That's good. No, I just wanted to just get your idea on this fuzzy complexes, right? So, I mean, it's well established now that that several complexes exist without the increase in the order, structural order, right?
Yeah.
So when they're complex, but my question is, I don't know about whether it's been reported in the literature. I mean, you've seen this Ben Schuler's work on protealpha and histone complexes, very high affinity complexes.
Yeah.
But what if such fuzzy complexes do exist in cytoplasm as well? And if they, I mean, they do, but why are they not susceptible for enzymatic hydrolysis? Because I'm imagining that you're going to have a fuzzy complex, which is much of more exposed regions susceptible for enzymatic catalysis, right? I mean, if they exist, I mean, do they exist in the compartmentalized zones that they're sort of protected from these, or how do they work in cytoplasm?
I think so. You have your answer in your question. So with very high probability they will be compartmentalized. So number one. So number two. So what it will be the best example of this fuzzy complex? Any membrane-less organelle.
Yeah, that's right.
So when it's formed, it's definitely based on this fuzzy interaction. So you don't have much of very specific binding over there. You have a lot of touching, but you don't have any specific connections. Yes.
Yeah.
But now it actually brings us into very, very interesting field. So why do we have formation of this fuzzy complex-based membrane-less organelles? So why do we have liquid-liquid phase separation? So one of explanations will be we have specific binding places in proteins which are involved in interaction. Yeah, it will be representing part of a story, but not entire story. So a complete story probably need to take into account water. And what I mean by that, we do know that protein structure depends on environment. So we have sequence which is able to fold or non-fold depending on environment. So which means that so we have undoubtedly, we do have a very strong effect of water on protein. So, but somehow we are forgetting that so it's not one-way road. So protein will have also very strong effect on water. And each protein will have specific my own water around me. So so phase separation probably, so one of basic mechanisms for phase separation will be finding like water. I like my water more, I like your water more, so we are staying together because we are in same water. So, and in this particular case, yeah, it could be some interactions in between proteins, so but mostly it will be going through water. So, and it also will give you an explanation of why maybe you will not find too many proteases inside of this organelle. So because water of proteases is not comparable with water of these membrane-less organelles, they're not coming in. So we do have this partitioning, so it will be partitioning out. So they are not going inside because they don't like it there.
Right. So the phase separation is the one of the common mechanisms by which
Yep.
they compartmentalize and then.
Yep.
Okay, great. Vladimir, so you covered a lot of topics, and I'm sure you did not have time to talk about many other topics. Would you like to share some of the key latest findings related to the protein aggregation and viral infection? Anything that you can share with this audience would be great.
Okay. What do you know about these viral factories?
Nothing. Start from zero.
Um, it goes back to membrane-less organelles and phase separation, liquid-liquid phase separation. So what many viruses I do, so when they are in host cell, so they will create specific compartments which will be entirely working for them. So it will be so-called viral factory. It's not aggregation, it's not pathological aggregation from viewpoint of forming amyloid fibrils. So, but it's a very crucial. It's not so can you call this stuff as aggregation? Maybe you can call this stuff as aggregation. Formation of this phase separated stuff place for virus to deal with. It will be one situation. Many viruses will affect biogenesis of different membraneless organelles in a cell. What virus actually need to do? So what is a major function of virus inside of cell? Is to kill everything which is not related to stuff which is needed for virus and convince everything which is working for virus to work for virus. So, and as a result, so you do have a lot of possibilities of how viruses can affect all of this stuff. So on top of that, I cannot exclude that some viruses will be related somehow to pathological aggregation from viewpoint of amyloid formation. So you know the stuff probably better than me. So people are discussing that so viral infection can be related to Alzheimer's disease. So now COVID stuff, people are saying you have one of likely outputs. You will have accelerated or you have worsened situation with neurodegeneration because of COVID. We are a highly balanced, highly resilient system. So, but we are resilient up to what particular degree, and if something is screwed to a very large degree, so then everything goes out of control. So, yeah, even if you just consider, even if you just consider these membraneless organelles, which are very, very hot topic now, they are crucial because they're responsible for very important set of regulations and mechanisms inside of cell. So, but they're transient. So many of them are transient. They're not existing for a long time. And they're definitely very crucial on one side, but they're also very dangerous on another side. So because now you have droplet with highly increased local concentration of protein, and definitely it will be a place for trouble. So if it's not dissociating in the right way, at the right place, so you do have your initiation of aggregation. So, and this could be actually one of mechanisms of how viruses can contribute to pathological aggregation and formation of fibrils.
Thank you. So there's some a lot of evidence for this, for the correlation between Alzheimer's and HIV infection.
Mm-hmm. Oh yeah.
A lot of studies are coming out on as well. So are there any other questions? Bikash, do you have any questions?
Yeah,
No,
I
go
have
ahead.
one question actually. Yeah, it's a very nice presentation. I enjoyed your talk. So I think in one of the slide, I don't remember the slide number because you don't have, you are trying to make the tree diagram for protein structure, function, and relationship, where you have shown like you started from the amino acid sequence. So can you go back to that slide if you have?
This one?
Yeah, you have the flow chart for
this or no?
No, you have the flow chart for protein structure, function, and relationship. Like you started from, yeah, this one. So here, no, I think the last slide, yeah, this slide. So here somehow I was wondering, like, I think like Dr. Vijay has already mentioned, like when you start from the amino acid sequence and we work through this kind of tree diagram, like the non-folding problem, like when you don't have non-folding, you know that like during the translation, your protein have some kind of non-structure, and then the chaperones plays a major role, then you get order, structure, and function. That is very known. But like when I look at the top one, like when you say your amino acid sequence have folding problem, and you go from directly a 3D structure to order and disorder, I am somehow lost here. When
No. So, okay, so it's a misconception. So folding problem, so its sequence is working on solving folding problem.
No, that I understand. I'm just trying.
So foldable protein is able to fold. Okay, so this is folding problem for a sequence. We have a structure.
Right, like, but in the amino acid sequence, like if you think about the nascent chain biology, like the co-translational modification, not the post-translational modification. So most of the IDBs are actually connected to the nascent chain biology. So like the unstructure happens right after the ribosome, like during the co-translational process. So in that context, if I look at your tree diagram, I would expect that the amino acid sequence, when you have a folding problem, you will get directly a disorder structure rather than getting a folding structure. And your tree diagram is well suited for P53, or let's say pre-melanoma, like P male, those like who have multi-domains, and you have some kind of domain structure and some kind of domain unstructure, and that make the function. For example, you mentioned about your virus fusion with the host and these kind of things, right? But how does it fit when you look at, for example, like alpha-synuclein? Because alpha-synuclein is itself is disorder. It doesn't have a structure starting from the co-translation. So this tree diagram fits there.
So it will be over there. So instead of going to 3D structure, it will be going into conformational ensemble. So we have, depending on sequence, we have two possibilities. Some sequences can fold, and some sequences cannot fold. So, and instead of one structure, we have a flexible ensemble, and we have majority of functions coming from this ensemble.
Yeah, but in this case, like when you go in the downward, like the disorder to order, so the orderness in the alpha-synuclein comes like when they form some kind of self-assembly or they interact with the membrane, they don't.
Sure.
So
why protein folds to start with? So why foldable protein folds?
Yeah, they to do the function like with the function.
No. It folds for, but why? What is the mechanism of folding?
So there are several.
Mechanism of folding is related to specific set of sequences, amino acids in a sequence. Yes. So we do have the so-called folding code.
Right.
So we have sequence which somehow knows how to fold under physiological conditions. Okay.
Right.
So, and therefore, I'm talking about solving folding problem here. So protein knows how to fold under these conditions. So now you have other set of sequences, so which don't have all information for folding. So which means that you don't have everything which is needed for this protein to fold independently. So, but you might have some pieces of this protein, so which potentially can fold if they will find a partner. So you can imagine a situation, you have a short disordered region or structured region located in low disordered piece. Can this short region fold? Even from viewpoint of very simple physics, it cannot. So because you have very strong pulling forces coming from disordered tails. So, but now if this piece will be able to find a binding partner, will interact with it. So now you are in good shape, and this stuff can be done.
Yeah, that's true. Yeah, that's true. That is understandable. But I'm thinking in terms of like, because you said that non-folding problem. So I would assume that most of the nascent chain biology problems doesn't fall under the non-folding problem. They fall under the like some kind of problem with the folding. So in that context, I was somehow like lost using this tree diagram. But I think like your explanation makes sense. Like the folding could be happen, that depends on many factors as you mentioned. It could be a chemical chaperone, it could be a like protein chaperone, it could be self-assembling, it could be some kind of like factor. So there are so many things. But like the only question I was wondering, like whether you are trying to tell that whether the nascent chain biology that we know is actually falling in the folding problem, in the non-folding problem. That is where I was lost.
No, I think it depends on the protein once again. So if protein at the end will be folded, so then your nonsense biology will be in folding problem. If your protein is not folded, it's non-folding problem.
Okay. Yeah.
So unfortunately, there is no uniform answer to this stuff. So yeah.
All right. Okay.
Okay. Okay. Let's thank Vladimir for the wonderful talk and for the great discussions. We all enjoyed it very much. And thanks to you, and thanks to all the speakers, and thanks to all the participants who really gave us a lot of insights into the topics in this year. So wish you all happy holidays and a very happy new year.
Thank you, Rams. Thank you all.
Yeah.
Thank you, Vlad.
Yeah, thank you.
Thank you, Vlad. I really enjoyed it. It's really interesting and inspiring. So
thank you. I hope that you are not just trying to please me and say me good words.
No. Yeah, I actually had. Can I ask my last question about LLPs and the viruses? Have you looked in the viral proteins? What is the abundance of IDPs? And whether the viruses are bringing more the stickers or the linkers, you know, when there are these two components for the LP formation.
Yeah.
It's kind of
so this is disorder prediction for viruses, and as you can see, viruses are very diverse. So most ordered proteins will be viruses, and most disordered proteins will be viruses. So depending on how they're using disorders, so this will be kind of in this way. Now related to liquid-liquid phase transitions, I think it depends on the protein, on the virus.
Okay.
I'm not sure that all viruses will have viral factories. So stuff which I mentioned, this viral factories, it's phase separated place in the cell on which virus will be assembled. So then they were described for maybe about 10 viruses. I'm not sure that they were described for all viruses. So this is what I know about it.
Yeah, but yeah, that's true. But as you were saying, the things might start popping out, because even like encapsulating the viral particle, if you think of it, it's like phase transition needs to require almost like.
So do you know when phase separation was described in the cell for the first time?
No.
Can you guess?
I don't know, in the early this century, I don't know, 20 years ago.
Yeah, this is what we believe. So this stuff was described for the first time early this century. Yeah, maybe not even early this century, maybe about 10 years ago, okay? So, but in reality, first stuff was published in 1765.
What is it on material system or polymers or?
No. I'm talking about biological.
Is it a nucleolus Cahal bodies?
So nucleolus. So it was first discovery, but also people were observing this magic behavior. If you're treating a cell with some stress, so they start to be opalescent. So you do have formation of something inside, so which is transient. So exists for some time, but then it's gone when stress is gone. So, but then we are not able to, we are reductionists, yes? So if you have complex stuff, we try to pull it apart and see how the stuff works. So we discovered nucleus because we are able to take it out. We discovered mitochondria because we are able to take it out. So when you have membrane-less organelle, it's a phase transition. So what does it mean, phase transition? It's like this. So you have changes, you have phase transition, you have changes, they're gone. You are trying to open cell, majority of your membraneless organelles will be gone. So 10 years ago, something around that time, a correlation was found in between appearance of something strange related to cancer. So, and this actually brought attention to these membraneless organelles. So how many of those? I have a review published a couple of years ago, and we listed about 36 different organelles over there. In reality, I think it's much more. So now we are starting to pay more attention to it. We will be seeing that. So this stuff is everywhere. So
interesting.
Yeah. Thank you. That's really exciting. And it's again, it's, if you think of it, phase separation is is something which, if you find the right set of conditions, all the proteins will.
And Rams, you absolutely,
What? Did you say something?
Or,
No, I didn't call you.
Can you hear me?
Yeah, I can hear you. Yes. Oh, he's gone. No.
Yeah,
He's coming back.
He'll come back.
I think we should ask him to come and attend the ZOOMinars. He's like a library. He knows a lot of stuff.
Oh yeah, that's
There's a lot to learn from him.
Yeah.
Vladimir, can you hear us? We're able to see you. Maybe you're frozen.
Yeah, I think his internet is a little bit off. Well, I have to leave, but Rams, Magda, and Bikash, I think excellent. I mean, I think I enjoyed the ZOOMinar in this semester super well. So it was fantastic.
We are thankful to the participants that were able to break the barrier and keep the education continuing.
Yeah.
Despite the challenging situation. That was the idea we started with, but the participants gave us a lot more.
Oh, that's a brilliant idea. Well done also. I enjoyed. Thank you, guys.
Yeah, you're welcome.
Vlad, I'll get back to you on the granulin part. I have to write something to you. And I owe something to Rams as well. So.
As I told you, if something will visit my head, I will let you know.
I would love to have that open head than mine. So, see ya.
Bye.
Bye-bye.
Bye. Bye.
Vladio, I have a question for you. So could you, we have the ZOOMinar scheduled all the way to end of June, end of June next year. May we request you to kindly come and attend every ZOOMinar, because you are like a library, you know a lot of stuff. So you can be by default participant to answer some of the questions or provide.
I cannot promise that I will be able to do it each time, but I'll try. So
what can we do to make you attend all the seminars?
It's just
can we bribe you?
It's not you, it's timing.
Yeah.
So if I'm free, I will try to attend. Yeah, for sure.
Yeah.
And can I ask you, as organizers, send reminders to me? So because
he's in the email,
I'll do that personally every time. Yeah.
No, because put him in the email list, automatically it will go on the previous night.
So okay.
So because we really need participants come, because these topics are very broad, interdisciplinary.
Yeah, absolutely.
Not everybody. And you give us a lot of topics, and very unfortunate that we could not hear more details on several of your topics. Maybe another time and another situation we can bring you in.
Do you know how much time do I need to talk about stuff which I want to talk about?
Now, you have the entire year, 2020, maybe entire century.
So I had a mini course about intrinsic disorder, so which are 7 lectures, 2 hours long.
Ooh.
So it's 14 hours, and I think I need more.
So if we make a dedicated IDP and just disorder alone topic, a focus symposium, would you be available to talk at length?
So at length meaning what? So 14 hours? Yeah, sure, of course.
We'll record it. We'll record it, and then we'll film it, and then we'll just digest it and make them available for people to understand.
No, I think it's a good idea, and I think it's necessary to do, because from my viewpoint, so you cannot talk about health or health issues without talking about intrinsic disorder. It's need to be there for sure.
So let me ask another question, maybe dumb question, maybe. So we are screening small molecule compound. We are screening conditions for potential way to treat or maybe intervene this disease, the pathology of the disease. So how do we deal with IDPs? Can we screen compound? What do you look for that? Maybe Christian is here. I wonder if Christian's compound works against IDPs aggregation to phase separate. Christian, if you want to come to the panel. Anyway, so maybe.
Rams,
so
he has to.
I'm sorry, what?
So Zhang, he has some question in the Q&A. Maybe he can directly talk because.
Yeah, you can promote him. You can come directly.
Hi, are you talking to me? This
Yep.
Go ahead, ask. Go ahead.
Hi, it's really very interesting talk and summarize a lot of questions that we have with IDPs. And specifically when you're talking about liquid-liquid phase separation, you said proteins have, you have your water, I have my water, and your water like my water. Something for that reason they form this phase separation. And it's very refreshing idea. And can you comment how about the aggregation? What's the function of water in forming aggregation?
So, but aggregation is drying out, yes? So aggregation is expelling water.
So that means you spell out your water and my water, so
you're squeezing out your water. So, for you, in order to form amyloid fibril, so with very high probability, you cannot do it. So starting from highly structured state, you need to be in more or less unfolded state. And I have a review with Tony Fink about this stuff in 2004. So you need to be highly disordered. What does it mean highly disordered? You're highly hydrated, so you have a lot of water inside. So and when aggregates start to form, so all of this internal water will be expelled. So you are drying out, so you're expelling water.
So
this will be largest difference in between formation of liquid-liquid phase separated liquid droplet and protein aggregate. So because in liquid droplet you still have a lot of water, and aggregate will expel a lot of water.
So can I understand like this, as you say, when you form phase separation, you still keep your own water.
Yeah.
A lot of outside water was taken away, right? And when you form, go ahead.
No, so when you have a droplet, so a droplet inside will have water which is different from bulk water. So yeah. And water inside of droplet so will be created by molecules which are located inside of this droplet. So you have your own water, you're bringing your own water to your partners, and you're creating own water in this droplet. So from this viewpoint, you do have a different water. Yeah, your bulk water is not there. This is right, yes.
I see. Okay, so can you define what's your water? What's each protein's water?
It's a good question, and answer is nowhere. So because people are not looking at this stuff in a very serious way, we are trying to do this work, and it's very difficult. So water is character. So okay, so let's start with very simple question. So so what is water? So what do you know about structure of water?
I think water is polarized molecules. So that's why each, let's say, lysine, positive charge, and glutamic acid, they have different kind of water organized around them.
Okay. So, but if you will exclude proteins, you will just look at bulk water. So what can you tell about structure of this water?
Okay, so water, they form intermolecular hydrogen bonding, and they could organize themselves in a way, but what way is very not, it's highly undefined.
Yeah, so but this is an answer to your question, because we don't know how bulk water looks like. We do have a very crude description of what is there. We can say, okay, we have a particular set of hydrogen bonding, and depending on whom you're talking to, people will be talking about either 4 different types of hydrogen bonding up to 35 different types of hydrogen bonding. And it's highly debatable. So it's a highly dynamic system. It's not existing as you can find some crystal-like structures inside of water, and people are discussing even for ice. You have different types of ice structure depending on how the stuff is organized. But now, if you will take into account that water is highly heterogeneous, what is going on when you will put something in water? So definitely this heterogeneity around something added, so will be different from bulk. So you pointed out very rightly, if you have charged residue, it will organize water around them in a specific way. And you can extend the stuff to entire protein, and you will see that you do have water which is quite different. So now question is how far from protein surface so this own water will be going out. And my honest question will be, I don't know. So because nobody was able to convince me that 2 layers of water will be sufficient, 10 layers of water molecules will be sufficient, or it will go further. So, but what is absolutely clear, so water around structured protein, so will be very different from water around disordered protein. Even from very simple, or very simple understanding. So ordered protein is compact, which means that so you